A protein is a chain of amino acids. The process of linking amino acids together to make a protein is called protein synthesis. Protein synthesis occurs in a cell's cytoplasm. mRNA leaves the nucleus through a nuclear pore after it is transcribed from DNA. The assembly of protein in the cytoplasm takes place on ribosomes. Ribosomes consist of two subunits, a small ribosomal subunit and a large ribosomal subunit. Floating within the cytoplasm are amino acids and transfer RNAs, or tRNAs. Each kind of amino acid binds to one specific type of tRNA. Notice the 3-base region on the tRNA. It is complementary to a 3-base region on the mRNA. Each group of 3 bases on the mRNA is called a codon. The complementary 3-base sequence on the tRNA is called the anticodon. A specific tRNA carrying the amino acid methionine binds to the mRNA. The anticodon on the tRNA matches the codon on the mRNA. A second tRNA with its attached amino acid binds to the second codon of the mRNA. The first amino acid is transferred from its tRNA to the second amino acid, and a bond between the two amino acids is formed. The protein chain has begun to grow. The first tRNA detaches from the mRNA. The ribosome moves down the mRNA to the third codon. This codon is recognized by the anticodon of another tRNA carrying an amino acid. The growing protein chain is transferred to the third amino acid. The second tRNA detaches. The ribosome again moves along the mRNA. A codon is exposed. Again, the codon is matched by the anticodon of a tRNA carrying an amino acid. The protein chain is shifted, adding another amino acid to the chain. The tRNA that gave up its amino acid detaches, and the ribosome moves down the mRNA strand to the next codon. This process, called elongation, builds proteins one amino acid at a time. The 3 bases that make up a codon determine which tRNA attaches. Since each tRNA carries a specific amino acid, the sequence of amino acids in the new protein is determined by the sequence of bases in the mRNA. The ribosome continues to move along the mRNA. At the end of the mRNA, the ribosome encounters a stop codon, such as UGA. The completed protein is released. The tRNA floats off, and the mRNA detaches from the ribosome. The ribosome splits into small and large subunits.